Chapter 2, Basic Microbiology. In this section, we are going to discuss basic microbiology and testing. The purpose of this chapter is to provide you a basic understanding of yeast, mold, spores, and bacteria. We'll discuss total yeast and mold counts and the different methods of how they are tested and counted. This section may not be necessary for everyone, but it is good to know a bit about what we are trying to kill with the RFX and have an understanding of how microbial testing works. Essentially, there are 4 different types of things to be aware of. These are yeast, mold spores, and bacteria. Yeast, mold, and mold spores are a type of fungi, which are quite different from bacteria. Yeast are microscopic single-celled organisms belonging to the fungi kingdom, a group that also includes mushrooms and mold. Yeasts are found throughout nature, inhabiting soil, vegetation, and aquatic ecosystems. They reproduce in moist environments, but can also live for thousands of years in a dormant state without water. Yeast will not reproduce on dry cannabis, but it can thrive on growing cannabis plants and wet cannabis, which means they are still present after being harvested and dried. Most yeasts are not harmful and can actually help the living plant in many ways. However, most laboratory testing does not differentiate between good yeast and bad yeast, so we need to get rid of all of them. If you ever had a chance to see the way testing lab counts yeast, they typically appear as small yellowish clusters. Mold is also a type of fungi, but unlike yeast, mold can grow into large clusters. In cannabis, it usually appears either late in the flowering cycle or during early drying when the plant loses its natural defenses. This is why it's important to dry cannabis quickly and reach a moisture level below 15%. However, looks can be deceiving. Cannabis that looks perfectly fine can still fail testing for levels of microbials. This is likely due to mold spores, which can be thought of like seeds, or it's because of mold that has not fully grown and it's still invisible to the naked eye. Did you ever wonder how little mold it takes to fail a test? You'd be surprised to know that 1 square centimeter of mold can produce up to 2 billion spores. That 1 square centimeter of mold contains enough spores to contaminate over 450 pounds of cannabis. Spores can come from cracks in cement floor, grow in soil, circulate in HVAC systems, and even from your clothing. Spores can literally wipe out your entire harvest. The RFX primarily targets mold spores and yeast, which are usually what cultivators have issues with. RF will kill growing mold, but when mold becomes visible, such as powdery mildew or botrytis, the damage is done and the product is past recovery. The RFX also will not remove aflatoxins, which are dangerous compounds formed by fungal metabolism. We do not recommend using the RFX to treat visually contaminated material as it may contain dangerous toxins and the RFX will not visually remove anything. Bacteria are another type of single-cell organism. They can reproduce rapidly and are responsible for many illnesses and infections in humans. A high bacterial count, measured as total aerobic microbial count, or TAMC, in cannabis is usually an indicator of poor cultivation or poor environmental conditions. Bacteria are more heat-resistant than yeast and mold. Salmonella and E. coli are rarely prevalent in cannabis. Nonetheless, they are required as part of regulatory testing due to their high safety risk to consumers. Now that we've discussed a bit about microbiology, we can start talking about how laboratories perform testing and how this applies to your business. In the food industry, there are federal standards all businesses and labs must comply with to ensure uniformity. However, in the cannabis industry, standards and testing methodologies vary by country, and in the US, where there are no federal guidelines, they differ from state to state. In short, cannabis testing is complicated and a little different for everyone depending on where your business is located. We will discuss the most common standards and testing methodologies. These are agar plating and PCR. The most common microbial test is TYMC, or total yeast and mold count. This is measured in colony-forming units per gram, or CFUs for short. The most common limit is 10,000 colony-forming units per gram. A colony-forming unit is 1 colony, or dot, or fuzzy spot on an agar plate. Generally speaking, the large fuzzy things are mold and the small circular dots are yeast. Each one of these spots, no matter how small, comprise a colony-forming unit. This plate appears to have about 250 colony-forming units. Now, imagine counting 500,000 of these. It would be impossible, which is why labs use a technique called serial dilution. Serial dilution is a way to dilute a sample to a manageable level for a person or computer to count. When you send a sample for microbial testing to the lab, they will usually take 1 gram, crush it up, and mix it with 9 grams of a buffer liquid. This forms a 1 to 10 dilution. They will then take 1 gram of that mixture and mix it with another 9 grams of buffer, forming a 1 to 100 dilution. Doing this again forms a 1 to 1000 dilution, and doing this again a 1 to 10,000 dilution. After making the dilutions, the lab will either perform PCR testing or agar plate testing. Agar plate testing is the standard method of testing for most agricultural and pharmaceutical products. This method only counts live, viable organisms that will grow on plates. Many microorganisms will not grow on an agar plate because it isn't suitable to promote growth. It's a bit like expecting every plant on Earth to grow in the same soil, with the same nutrients, with the same pH. For instance, powdery mildew is notoriously impossible to grow on an agar plate, and thus will not be captured in the test results. Flawed as this may be, it's the standard for the industry. Agar plates are essentially a gelatinous broth of nutrients made suitable for the widest possible range of yeasts and molds to grow. There's also a gelatinous broth of nutrients made suitable for bacteria or specific pathogens that labs also use to count bacteria. Usually, a lab makes several plates of the same sample to determine the total yeast and mold count. They do this by plating 1 milliliter of each serial dilution and incubating it for 5 days. If you ever wondered why it takes so long to get your test results back, now you know. This means the lab will plate a 1 to 100, a 1 to 1000, and a 1 to 10,000 sample to determine the total yeast and mold count. Usually, a lab will average these results together to arrive at the result. To illustrate how this works, 10 spots of growth on the 1 to 1000 dilution plate would equate to 10,000 colony-forming units per gram. Sometimes a lab result will simply say fail, or TNTC. This essentially means even on the highest dilution, the sample was too numerous to count. When possible, it is always beneficial to know the actual colony-forming units per gram from your lab. You'll just have to request the lab to do the necessary serial dilutions. PCR testing is a method that uses DNA sequencing to determine the total amount of organisms present in a sample. There are many different types of PCR testing methodologies, but they all essentially use a series of reagents or chemicals that amplify the DNA, which is then counted by a computer. This is a gross oversimplification of the process, but what is important to know is that there are 2 main types of PCR. One type counts all foreign DNA, alive or dead. This means any type of remediation will be read as ineffective, as both the dead organisms will be counted as well as the remaining live organisms. New advances in PCR technology now allow for the counting of live DNA only. However, there are certain microorganisms that show up in PCR testing that do not readily grow on agar plates. Just to add more uncertainty into the equation, testing in the cannabis industry suffers from a lack of standardization. Some states allow multiple different testing methodologies, and nearly every state has different rules from one to the other. If you have a choice in how your product is tested, we recommend using the tried and true test of agar plating. Passing microbial testing depends on which test your regulatory body requires. These are the most common tests, any of which may be required to pass. The bacteria E. coli and Salmonella, bile-tolerant gram-negative bacteria, TYMC or total yeast and mold count, TAMC or total aerobic microbial count, coliforms, and aspergillus. Okay, that's it for your primer on microbiology and testing. For the remainder of this course, we will refer to TYMC, bacteria, and aspergillus collectively as the microbial load.